Hello again, broadcasting from the back corner. We move this out. You remember this diagram? I'm sure you do. Well, at least you remember that you've seen it before. Take a minute and see if you can remember what goes where. Which number is indicating the blood vessel? Which one's indicating the bone and the tendon? That was always a race to come up and fill in because that was easy. But the rest of them, that's the challenge. We're going to talk today about muscle contraction. We're going to get into sliding filament theory just a little bit. But first, by way of review, you remember where those go? Number 3 is talking about, of course, the big bundle. Number 6 is indicating the little bundle. Pause it so that you can fill those in. I'm going to keep going because I can. So that's filled in. So the muscle fiber sticks out. The muscle fiber is part of a larger bundle, a fascicle, which is wrapped in paramecium. The blood vessels right there. The epimecium surrounds the fascicles and holds them together. The endomecium surrounds the individual muscle fiber. So it's fiber, endomecium, fascicle, paramecium, muscle, endomecium. Bring it all down together into a tendon, hook it onto a bone, and now it's anchored and you are ready to move. You might remember this diagram. It's in your note packet. This is all from the note packet. I didn't add anything this time. I took a few things out to streamline it. If you haven't found in the note packet where we're at yet, pause, flip there. I'll wait. So here you can see we've got the bone to the tendon to the epimecium, the muscle bundle to the fascicles, to the muscle fiber. We stretch that fiber out. You can see the myofibrils inside the fiber, the nuclei on the outside of the fiber. We're talking about skeletal muscle. One of the traits of skeletal muscle was that they were multinucleated. You can see the blood vessel. You can see the sarcolemma, the cell membrane around the myofibril. And you can see on this zoom in right here, we have a myosin filament, we have an actin filament, and we have the individual sarcomere pieces. Zoom in on that. And you can see, again, we have the band here, the Z discs, which separate the sarcomere. We have the I bands, we have the A band. We'll get into all that. You made a flip grid explaining this, so it should be review, should be easy. Go ahead and pause it for a second. I'll slide out of the shot if you don't want my face on the screen. See if you can name all those parts. All right, so if you were playing along with us, or with me, and with everybody else who's watching this video independently in isolation, hopefully you were able to fill all those in. I'm not going to take the time to drag them around. I already did that. So the Z discs are the sides. The actin is connected to the Z disc. The myosin is between. So if this is my actin and this is my myosin, they're going to overlap like that. That area of overlap is the A band. Now, how did we say to remember that? We said that you have the Z disc on the sides. Then you have the I band. 2 I bands, 2 I's, 2 I bands. Then you have the A band, which is the wide band in the middle. Some of us have a wider A band than others. I inherited mine, honestly. Right in the middle is the H zone. The H zone is just myosin. The M line is the middle line between the Z discs. It's not a feature that you see on looking at a muscle fiber, but it marks the middle. It gives us a reference point. Why does all that matter? Because this is all going to work together, and this is working together like an accordion every time you move. It's amazing how much has to happen for you to be able to move, and yet it's like that. We don't even think about it consciously. So the example that is often used is this idea of a burning twig. Wait a minute. What is a burning twig? I mean, fire is cool. We like fire. Ooh, fire. But how does this connect to muscle movement? That's my job to explain. So muscle contraction occurs when you get a signal from the motor neuron. So you have a nerve that comes in, and that nerve is responsible for telling a group of muscle cells, time to move. You need to, there's the camera over there. You need to contract. You need to relax so that contract. Okay, now relax so these can contract and go back. Flex, extend, agonist, antagonist. Agonist pulling down, antagonist saying, not so fast, slow down. All these wonderful words. It really is its own language. So what happens is the nerve impulse travels down, talked about nerves and action potentials already. It travels down the nerve, it gets to the junction between the myo, the muscle, and the nerve, triggers that reaction, calcium comes down, and the neurotransmitter, acetylcholine, is released. Acetylcholine bounces across, fuses to the membrane around the sarcomere, the sarcolemma. Wow, these names go together. And that triggers our sodium-potassium pump. Sodium goes in, potassium comes out. That alters the electrical potential, action potential spreads down the sarcolemma, and the muscle cells in the motor unit contract. You don't have a nerve for every single sarcomere. But that chemical change, that acetylcholine spreading out, triggering the sodium-potassium pump, triggering the contraction, opening up the little tubules inside the sarcomere that we haven't talked about that release calcium, that all happens, again, all at once, really quickly, to trigger and to allow that contraction to occur. It's like, here's the connection, having a little twig. Now, if you've ever played with fire, you should be careful and responsible. I still have all my fingers. I was a good Boy Scout. I only burned things when I could get away with it. You take the match, you hold the match to the end of the twig, and the flame will spread quickly down the twig. That's the action potential spreading to all of the sarcomeres in the motor unit, and that signal getting there and saying, hey, we got to move. You need to contract. That brings us to sliding filament theory. So we've had this happen. We've dumped now. Let me slide out of the way. Look, I can move myself. We've got the, right in here. Don't point that. Doesn't do any good. So the muscle at rest, you have the actin filaments. You have the myosin filament. The myosin filament has these little golf clubs. And the protein complexes here on the actin filament, it's all at rest. Life is good, everything's fine. Then action potential comes. Sound the alarm. Gotta move, gotta move, gotta move. Here comes the calcium. Calcium comes in, attaches to the protein complex, which is made of two proteins, troponin and tropomyosin. Comes in, binds to those, and changes their shape. And it exposes, oh, look, there's little notches there that the golf clubs fit in. The calcium release exposes the binding site for the myosin, and the myosin can attach to the actin. These little connections here, this is important, these little connections are called cross bridges. That is the next slide. So this is the cross bridge. Once you have the cross bridge, now the myosin says, oh, hey, guess what? I've got some ATP. I've got some energy. I've bonded. It's time to move. Power stroke. The power stroke is the movement. So cross bridge, grabs a hold, power stroke, move, detach, got to let go, to reset, to grab, to power stroke. It's like a handshake. You have the cross bridge, the initial handshake, cross bridge, power stroke, detachment, reactivation. You move to the next person, cross bridge, power stroke, detachment, reactivation. If we were in class, I would walk around the room doing this. We can't do that anymore because contagious, we'd have to glove first probably. But we would do this to illustrate these steps. You've got this in your notes. It goes through the whole process. You've got this picture, but this is where I was going to spend more of our time in this video. And let me fly up there. Woo. All right. So the myosin head attaches to the actin, forming a cross bridge. We just said that. At this point, when this happens, this breaks ATP. So now you have an ADP, ATP, adenosine triphosphate, ADP, adenosine diphosphate. Where's the extra P? PI, inorganic phosphate. It's been detached. It's going to leave because it's got to go back to the mitochondria to get attached to a new ADP to make ATP to come back out and do more work. You mean there's even tons of stuff? We are just scratching the surface. So when that is released, that myosin head power strokes and it pulls the actin. I have a great illustration for this that I'm saving for the end. But basically, it's like you grab a rope, you pull the rope, you grab farther down the rope, you pull again. And as that happens, it's pulling, the myosin is pulling the actin is contracting, the Z discs are getting closer together, all the way down the chain of sarcomeres, and the entire muscle shortens and moves the bone. Which part moves? The insertion. Remember, the origin is the part that doesn't go anywhere. The origin is the part that doesn't go anywhere. The inserted end gets pulled. I'm just going to leave that. You laugh at the fact that I'm stumbling in my videos. I get to chuckle at some of your flip grids. So now we've got detachment. So the myosin head now has pivoted. It releases the ADP. It detaches and it grabs a new ATP. It uses that ATP energy to reactivate itself, resets, cross bridges and loses the phosphate, power stroke, loses the ATP, detaches, grabs a new ATP, resets itself, breaks that into ADP and P, and it's ready to go again. Cross bridge, power stroke, detachment, reactivation. So if you've got your fist here, if this is the actin, cross bridge, power stroke, detachment, reactivation, power stroke, cross bridge, detachment, reactivation, power stroke, cross bridge, detachment, reactivation, power stroke, cross bridge, detachment, reactivation. The important thing is the detachment, because it's got to detach to relax so that the muscle on the other side, as it goes through its cycle, the muscle sarcomeres on the opposing side, the antagonist side, are allowed to stretch back out. If the entire muscle moves, well, then you have a muscle contraction and you have movement. But if just one neuron fires at just one little bit of the muscle, that's how you get a twitch or a muscle spasm. Just a small part moves. You look down at your arm, and the muscle in your little part of your arm is going. there's a nerve there that thinks, hey guys, we got to be doing something. And all the other nerves around it are like, no, we don't need to move. No, come on, we got to move, we got to move, we got to move. No, knock it off. And eventually the twitch stops. The nerve gives up. It's like, okay, I guess I got a mixed message from somewhere. All right, so here's our steps. In the crossbridge step, the calcium binds to the actin, moves the troponin out of the way, exposes the attachment site, the myosin attaches to the actin. Actin, myosin, attach. Power stroke, myosin heads move, ADP is released. Detachment, ATP attaches to the myosin and it relaxes. Too far. Okay. Reactivation breaks the new ATP to get ready for the next cycle. Crossbridge, power stroke, detachment, reactivation, crossbridge, power stroke, detachment, reactivation, crossbridge, power stroke, detachment, reactivation. As long as you've got enough calcium and enough ATP available, the cycle is going to continue. The muscle is going to continue to contract until either it runs out of supplies or the message stops from the nerve, says, okay, that's enough. And remember that for every movement, you're balancing muscles on both sides. So if I've got my arm, the nerves in the bicep are saying, contract. And others say, okay, relax. And the muscles in the tricep are saying, it's our turn. So one side is contracting and the other side is relaxing. So in the contraction here, I've got my prime mover and my antagonist. There are a number of fixator muscles in the rotator cuff that are keeping the shoulder stable. And there are other synergists besides the bicep and the tricep that are helping with one or the other directions of movement. All muscles are complicated. We really wanted to be able to do this in person, which is why we jumped out and did brains. But since we can't do it in person, we do want to finish the muscles. All right, so here I have, let me move again. You have this page, crossbridge, detachment, power stroke, reactivation. Your job is to figure out where they go. Well, that's fairly easy, right? Crossbridge is where it connects. Power stroke is where it moves. Detachment is where it lets go, but it hasn't cocked itself back to grab again. Reactivation, now we're ready to make that next grab. Okay. Questions? Leave them in, there should be a comment box below, or you can email me. All right, so there's a couple of links here that are in your video, in your note packet. Sorry, keep track of what I'm doing here. There is also a document that is posted on Canvas that's called, what did we call it? We called it Videos to Watch for Muscles. And there is a whole long list of videos that are great resources. These two have been added to that list. And I want to show you one more thing to wrap this up. Because when I was thinking of how to explain the actin and the myosin pulling on each other, I went straight to Les Mis, specifically to the movie. Now, you can find the opening scene on YouTube. This may very well be a federal crime. I don't know. But it's out there in public on YouTube, and I'm going to show you a piece of it, and I'm going to talk over it a little bit. I apologize. Cardinal sin. I apologize to the fans of musicals. But that's what we're going to do. Let me make this nice and big. All right. And let me turn off the lights here in the studio. All right. Not that that'll make a difference, but at least I'm not quite as bright as in the background. All right. So I think I can play this. All right. So the important thing here to notice, if the ship is the Z disc, look down here. You got all of these ropes. Those are the actin filaments. And as we come down in here, we have the prison gang playing tugboat. These guys are the myosin. Now, there aren't waves crashing over your muscles, thank goodness. But they power stroke, detachment, reactivation, cross bridge, power stroke, detachment, reactivation, cross bridge, power stroke. Now, we're not going to keep going long enough for them to break into song, but they're the myosin heads. They're pulling on the actin filaments. They're making it move. At least it worked in my head. Hopefully it works in yours. Hopefully some of this somewhere will work in yours. But as all of this happens, that's how we get movement. From the origin, the muscle pulls on the insertion, and as those sliding filaments work along and do their job, we have movement. And then they have to relax, and the muscle back here will move to go back to the original position.